talk about it. Let us talk about the ABO system. And while speaking about it, we're going to be speaking about the formation of the A, B, and H red blood cell antigen. Because on the surface of the cells, we have antigens. In circulation, we have antibodies. So we're going to consider it to be the formation of the ABH antigen, which is a result of an interaction between three separate loci: the ABO, the HH, capital H, lowercase h, and SE, capital S, and lowercase e. So these genes don't actually code for the production of the antigen, but what it does is, guess, it produces a special or specific glycosyl transferase that adds sugar to the base precursor substance. All right, so in this interaction, we can have an appreciation that sugars are involved in the formation of the ABH on the red blood cell. So a paraglobulicide or a glycan is the same basic precursor material from which the A, B, and H antigens all originate. So all these antigens originate from these glycans. So specific enzyme transferase elicited by an inherited genes attaches to a paraglobin side or a glycan, and when this happens, we have the different resulting ABH antigen being expressed on the cell. So when terminal galactose on the precursor substance is attached to N-acetyl glucosamine in beta 1, there is a link to there's a 4-link attachment that is taking place. Right? The precursor substance on erythrocyte is referred to as the type 2. So beta 1 move into giving us a 4 linkage, and then that leads us to type 2. So let us see if we can put this on our board. So we have beta, BETA, beta 1 goes to give us a 4 linkage, and then this now gives us type 2. All right, so the ABH antigen on the red blood cell are constructed on oligosaccharide chains of the type 2 precursor substance. So if we have type 2, I hope that you are thinking there must be a type 1. So type 1 would come before type 2. So let us get into type 1. A type 1 precursor is referred to as the beta 1, and this has 3 linkage between the galactose and the N-acetyl glucosamine. All right, and this generally gives us another type of formation. So let us just put what we have stated. So we have type 1 and we have type 2. So type 1 is what we're talking about right at this very moment. So for type 1 we're gonna have beta 1, which is going to link to 3. So we have 3 linkages. So 3, beta 1, and type 1. So for type 2, as we discussed before, we have beta 1, same beta 1, but this has 4 linkages. So I want you to observe. So 2, 1, 4 rather, 1 and 2. Alright? So this is basically type 1 and type 2. So the H antigen is a precursor structure on which A and B antigens are made. So in order for A or B to be made, we need the H antigen. And that is why the ABH system of antigen is important for us to have an appreciation of their formation. So, inherited based on our inheritance of the H gene, this will result in the formation of the H antigen. So you could have FUT1H or FUT2SE gene. Alright? So these two genes are closely linked and located on chromosome 19. So let us put that down so we can have a visual as to what does that mean. So we have FUT1H and FUT2SE. Alright? And these are on the chromosome 19. So just remember that, alright? In contrast to the ABO genes, so when we have the ABO genes, we discussed previously that this was located on chromosome 9. Alright? So the H and SE are located on chromosome 19, while the ABO genes are located on chromosome 9. So the H and SE genes are not a part of the ABO system. Alright, so we need to get that clear. Yet there is the inheritance influence to the A and the B expression. So though they are not directly associated, they do indirectly influence the formation of the ABO antigens on the red blood cell. Then this brings us to another idea that we need to be mindful of. All right, and we think about the SE genes. When we talk about the SE gene, which must be inherited to form the ABO in the secretions. I want you to get the two things. SE, secretions. When we think about secretions, we are thinking about secretors. So just bear that in mind as we traverse the exploration of the ABH formation. So the ABH antigen develops early in fetal life, but it does not increase much in strength during the gestational period. So the RBC of the newborn has an estimated carrying capacity of about 25 to 50% of the number of antigen sites found on an adult red blood cell. So this is basically giving us the idea that when we're doing a reaction, there is a possibility that an infant will have a lower reaction when we're grading it, like 1 plus agglutination, 2 plus, 3 plus, 4 plus. In the adults, you may expect a 4 plus or a 3 plus. For the infant, you may expect a 3 plus, a 2 plus, or even a 1 plus based on the amount that is being expressed. All right, so just bear that concept in mind so that this will give you a guide or give you insight as to how different markers are expressed, and we can take advantage of this when we're doing our ABO discrepancy assessment. So this is important for us to know, and now that you know, let us move along. So consequently, reaction of the newborn red blood cell with ABO reagent antiserum are frequently weaker. All right. The expression of A and B antigen on the red blood cell only fully develop at about 2 to 4 years of age. And when it is fully developed at that time, it remains constant throughout the rest of the life. However, in some instances, when a person becomes a geriatric, the body starts to produce less antibodies. The forward will remain constant. The reverse, which developed later based on the antibody expression, because for the babies, as we discussed before, we don't perform the reverse blood grouping. So too, when we go back into a geriatric state, what happens is that the body's ability to produce antibodies is reduced, and this reduction gives us lower grading or difficulty in identifying the antibodies because the titer is so low. All right, so you were in as a child, you will be when you become a geriatric. So it's the same kind of systematic processes that take place. So in addition to this, we need to understand that the phenotype expression of the ABH may vary with race, genetic interactions, and disease state. I know that when we speak about race, many persons are apprehensive to disclose their identity, but when it comes on to the medical field, understanding where a person is derived from, the race of the person, their genetic markers, their disease state helps us as practitioners to come to a realization as to what are the possible causes of the condition which impales the individual. So when you're asked about your race, don't feel apprehensive or hesitant to disclose it appropriately. All right. Now let us look at the glycosyl transferase and the dominant are the immunodominant sugars responsible for the H, A, and B antigen specificity. All right. So let us put the gene, Gene, all right. Then we're going to look at the glyco, GLYCOSYL, transferase, all right. Then we're going to have, maybe I shouldn't write this out. Let me make some more glyco SYL. We're going to put transferase down the bottom, but this is one word, transferase. All right, but that is one word. Then we're going to talk about the immunodominant sugar, immunodominant sugar, and then we're going to talk about the antigen, ANTIGEN. All right, I hope that we can make this out. So we're going to talk about the antigen. Let's write that, ANTIGEN, TIGEN. All right, let us draw a line, draw a line, draw a line. All right. So the first we know is the ABH system that we're talking about. So let us talk about the H. When we talk about H, we are thinking the gene FUT1, right? So in order to get FUT1, the glycosyl transferase that we need is well, let's start with the alpha. It's alpha 2L, and it is fructosyl transferase. FUCOSIL transferase. All right, and then this here, the immunodominant sugar would be the fructose. All right, so don't forget the L that we have, so it should be L fructose, FUCO fructose. All right, and then this here would give us the gene, so the antigen that is going to be expressed is H. Simple. So that is the hardest one. Now for the easier ones, A. If a person is A, they're going to have alpha, and this is going to be 3, and not only 3, 3N. And then now we have acetyl galactosamine transferase, or some may say acetyl galactosaminal transferase. All right, so anyhow you want to pronounce it, as is applicable in your jurisdiction, but what I'm going to do is spell it out so that you can have it documented. So it is alpha 3N ACETYLGALCTOSAMINYL TRANSFERASE. All right, so That is it. So when we're moving to the immunodominant sugar, we have an N here. So we're going to put by the N, and the sugar is acetyl D-galactosamine. All right. So it's N, and we use the same name. And remember, we started with A, so we are going to end with A. Beautiful. Now, let's go to B. I don't want you to be intimidated by the glycosyl transferase. I just want you to be able to identify them and understand the sequence of operation. All right, so you can know the immunodominant sugar, which are easier to pronounce, enunciate, and you can know the antigen because those are simple. So it's based on the ABC free sequence, but instead of ABO, we have ABH. All right, so now we move into B. So B is going to be alpha, and just like the A, it's going to be 3. However, when it comes on to the letter, it's not going to be L or N, it's going to be D. And for B, it's going to be galactosyl transferase. All right, so galactosyltransferase. All right, so we're gonna be putting the immunodominant sugar, and remember the D, we put back that D, and it's galactosyl. Where does it come from? Galactose. All right, so we're going to write galactose, and then this, now we started with B, so the antigen that we expect to be expressed is B. Beautiful. So that there is exactly how the operation and the association with the glycosyl transferase and immunodominant sugar are expressed. So this is just something that we need to be aware of so that we can identify, and then we can explain as is needed. All right, so what I want you to know is that there's an interaction with the HH, capital H, lowercase h, and the ABO genes. So individuals who are blood group O inherit at least one FU1H genes, or its genotype HH, capital H, or H, capital H, lowercase h, and 2 O genes, because you know that to express O, you have to be OO. All right, so if you are AO, you're not going to express O, you're going to express A, but you can transfer the O, and you can also transfer the A to the child that is going to be derived. All right, so just bear those concepts in mind. So the H genes elicit the production of an enzyme called alpha 2L fructosyl transferase that is that transfers the sugar L-fructose to an oligosaccharide chain on the terminal galactose of the type 2 chain. So basically, this is explaining to us the interaction that takes place and how the ABO system interacts with the H system, so as we can have a mental visual picture. Therefore, let us consider the L-fructose is a sugar that is responsible for H specificity. The O genes at the ABO loci is sometimes referred to as an amorphous and does not elicit the production of a catalytically active polypeptide transferase. Therefore, the H substance remain unmodified. All right, so this is what we need to understand for the O portion of the blood group system. There is a O lookalike that is referred to as the Bombay. We're going to have to discuss that a little bit further so that you can be able to differentiate genotypically O individual in comparison to a Bombay individual. All right. The H genes is present on 99.99% of random population. Its allele lowercase h is quite rare, and the genotype HH, which is lowercase h, is extremely rare, and this is where we get the Bombay term to be applicable. So the term Bombay has been used to refer to the phenotype that lacks the normal expression of the ABH antigen because of the heredity or the inheritance of the lowercase h genotype. So the lowercase h genotype does not elicit the production of alpha 2L fructosyl transferase. All right. So accordingly, the L-fructose is not added to the type 2 chain, and the A substance is not expressed on the red blood cells. So we have no H being expressed. And you know that we need the H to give us the A or the P. So when that is not being expressed, then we have a problem. It is a problem because majority of the population doesn't have the Bombay type. Because majority of the population, 99.99%, has a capital H somewhere. So they have capital H or capital H lowercase h. The lowercase h is the Bombay type, which is the 0.1% of the population. All right. So when we are thinking about this, it's a problem because not that we can't identify, the problem exists because when we are seeking after blood for these individuals, it is arduous, especially if we don't have a population pool of individuals who have this blood group. So even though the Bombay HH individual may inherit the ABO gene, the normal expression as a result in the formation of the ABH antigen does not occur. It does not occur. So this individual, the Bombay, is phenotype OH, So that's O and subscript H. Let me just write it just in case you come across it. So it's O and subscript H. This here is the BAM. No, it's actually Bombay. Bombay. So it's BOMBAY. All right. And the expression is HH. So this is not blood group O. I want you to remember this. If you see this, this is not blood group O. It looks like O. It's observed in the forward as O, but this is not O. All right. Now, in the formation of blood group A, what happens is that the A gene, and you know that we stated that if a person is A, they have the A gene, they can be AA or AO. So this situation codes for the alpha 3 N-acetyl galactosamine transferase, which transfers the N-acetyl D-galactosamine sugar to the H substance. So when this transfer is done, the sugar confers A specificity. Simple as that. So all the information that we had previously is now coming back full circle. So we are seeing how it plays out. So the A gene tends to elicit a higher concentration of transferase than the B genes, leading to conversion of nearly all the H antigen on the red blood cell to A antigen sites, as many as 810,000 compared to 1,170,000 antigen sites exists on an A1 adult red blood cell in response to inherited gene. So we're going through this. I want you to also be thinking in your mind, when a person is A, we realize that they have more expression of A than when a person is B. So let us picture in our mind a person is AB. If we don't have AB available, the first alternative, if we're going to choose between A or B to give to the person, is going to be the one that is mostly expressed, which would be the A. And because A is usually predominant in most population, A would be more available for the transfusion process. So these are things that I want you to put together in your mind so as you can have an appreciation for the whole flow of things. Now let us get to individuals with the blood group B. So this is inherited. An individual with B can be BB, capital B, or it can be BO. All right, so these genes code for alpha 3 D galactosyl transferase. All right, and this attaches the D galactose sugar to the H, and when this does, there is a situation that is placed or considered the type 2 precursor substance through which the action of H can generate the B. And that there is how we get, in essence, the B being expressed. This sugar is responsible for the B specificity, anywhere from 610,000 to 830,000 B antigen site exists on a B adult in response to the conversion of the H antigen by the alpha 3 D-galactosyl transferase to produce or produced by the B gene. So if you have the B gene, then you know which sugar was implicated in the expression. So let us go back now. When both A and B genes are inherited, the B enzyme seems to completely try its best to compete more efficiently for the H substances than the A enzyme. As a result of this enzyme competition, the average number of A antigen on the AB cell is approximately 600,000 sites compared to an average of 720,000 B antigen site. So the interaction of HH and ABO gene can be observed. So this is a good place to pause as we allow a moment in time to digest the information that has just been presented to us. As we have covered in brief the interaction of the H, capital H, lowercase h, and the ABO gene. So we have established the gene, we have established what is the glycosyl transferase, we have established the immunodominant sugar, and we have established what is expressed. We have also identified some of the potential sites expression in what quantity, so that we can have a mental picture or mental framework as to what would be possible for transfusion. So sometimes in transfusion, the thing that is given is not necessarily what is available, because if it's available and it is not compatible, then that becomes problematic. So what is normally being given is that which is suitable for the specific situation or the specific case, and we have to take into consideration the population expression as well, so that we can use that information of their race and the demographic information to determine what is their medical condition, the disease state, to determine what is the most suitable and viable product. So as we continue our journey through the immunohematology aspect of the profession of medical technology, medical laboratory science, clinical laboratory science, laboratory medicine, I hope that this will give you insight as to some of the theoretical frameworks that are used in the different investigation in coming to a conclusion as to what is most suitable in different situation and cases. With that said, thank you once again for spending some quality time with us. I hope that the information that you gained was meaningful, was impactful, and you have a better grasp and understanding. And with that said, class is dismissed. Have yourself a great day.